XEarPro, University of Trieste, and Aerosol Devices presents. Bioaerosol International Symposium.
Okay, so I think we can start. We have about 75 attendees, and the other will join us in the following minutes, but we are on time, so we can start. And it's a pleasure to give the word to the director of the Department of Chemical and Physics from the University of Trieste, Professor Stener. So please, if you want to start with your welcome. Thank you.
Thank you. So first of all, thank you very much for your invitation. So dear participants, speakers, and co-organizers, it's a real pleasure for me to open this symposium, which is a clear example of how public research institutions and international private technological companies can cooperate for developing and sharing knowledge, supporting the solution of relevant societal issues. We are here for sharing some pieces of knowledge that can be useful for characterizing biological components of environmental aerosols that are relevant for several scientific aspects, but now are important actually for a better understanding of the spread of COVID-19. As highlighted by the editorial of Nature paper of 2 February of this year, probably you know very well about this coronavirus in the air, there's too much focus on surfaces. Our department, so the Department of Chemical and Pharmaceutical Science of Trieste University, has been actively involved in supporting research on environmental presence of SARS-CoV-2 and in the assessment of materials used for personal protective equipment since early spring 2020. So at the very beginning of the emergency. And also with the stimulus and support of the rector, by our rector of the University of Trieste, with personal laboratories and mechanical workshops and so on. So it has been a real challenge for us, this. And we strengthened cooperation with colleagues from other departments, for example, Life Science Department, Medical Science Department, Engineering Department. In November, last November, we signed a formal scientific cooperation agreement with TCR Tecora, sister company of XEarPro, which is main organizer of this symposium. XEarPro brings here Professor Chang Yu Bu from Florida University, welcome, and Aerosol Devices, scientists and developers of what has been defined by top aerosol scientist Professor Jimenez, H-index 108, so-called an impressive technological advance. For example, the laminar flow condensation growth tube for sampling infectious virus from room A. We will listen to introductions to the issues reported in the title of the symposium by our environmental chemists and microbiologists, and to the presentation of these technological advances. So thank you to everybody, and enjoy the symposium. Thank you.
Thank you very much, Professor Stener, for your welcome. So please, Tanya, if you want to go on.
Okay, great. Welcome to the Bioaerosol Symposium on behalf of Aerosol Devices. Aerosol Devices is a small women-owned business headquartered in Colorado in the United States. Its founders, Pat Keaty, who will be featured here today, and Dr. Suzanne Herring, have dedicated their careers to aerosol research. They are both past presidents of the American Association for Aerosol Research. Since 2014, Aerosol Devices have been working on refining a commercial aerosol sampling portfolio with the crucial support of their research teams, as well as collaborations from various university scholars and research experts, such as those you're going to listen to today. We look forward to sharing about our technology, plus also the research tools, the research that our instruments have created. So thanks a lot, and look forward to getting to answer some questions and getting to hear the presentations. Thank you.
Hello to everybody, welcome. As XEarPro, we are the Italian distributor of Aerosol Devices. It's a proud for us. I'm personally proud to be here with all of you. We were around 200 people today connected, so this is a science, a positive science that it is an important thematics, and mainly in this period, or this bad period for everybody in Italy and worldwide. So thanks really to be here, and I wish you a good experience. And let me introduce you, Grazia. She is our senior scientist. She is also an expert in Bioaerosol thematics. And again, enjoy the meeting and good luck.
Okay, thank you, Marco. I share just some last information about the meeting. I show the screen just to remember the program. So this is the program for today, and we will have 5 presentations by international speakers. Each presentation will last about 15 minutes, and after each presentation, we will try to have time to answer your questions. So we hope to have your questions, your comments. Please all the attendees by using the Q&A function. In the Zoom, and be able to answer all your questions. We will try to end a symposium with a form in order to go on with the symposium. Later available for all the attendees. You will receive an email after this symposium with the link to see the recording of this symposium. And if you have problems with the connection, or if you have questions, if you have any issues, please you can write an email during the event to Eduardo or Marco, who are following the meeting, and they will try to help you in solving the issues. So it's a pleasure now to start with all of you, and I give the word to Aerosol Devices to start with the first speech. So please, Tanya, you can introduce the first speaker.
Okay.
I stop the sharing. Okay.
So while Pat's presenting her slides, getting her slides together, I'll present Pat Keaty. Pat Keaty is the CEO of Aerosol Devices. She's an entrepreneur with a passion for creating superior aerosol measurement capabilities. And it's really in the effort to improve environmental health, safety, and quality of life. She graduated from the University of Minnesota with a master's degree in mechanical engineering. Pat has developed her career in both commercial roles and research roles in many different companies and organizations. She's a past president of the American Association for Aerosol Research and a holder of 2 patents. Pat has been successful in converting the patented condensation growth tube technology into a series of commercial samplers that are used by leaders in the aerosol research community. So with that, I'll hand it across to Pat.
Thank you very much, Tanya, and for all of the welcoming committee. This is a great pleasure for me to be able to address you virtually. As was said, I am president of Aerosol Devices, and we are located in the Rocky Mountain West of the United States. And I've been involved in aerosol instrumentation my entire career, and never before have I seen such interest globally in aerosols. This has truly been quite the year. 2020 was quite the year for aerosol scientists. And I hope that 2021 will be the year where many of our unanswered questions are going to be addressed with regard to the airborne disease transmission. And aerosol scientists will continue to get the recognition that they so deserve. And now I'm going to see it. There we go. Okay. So aerosol transmission of the SARS-CoV-2 virus has been very controversial, and it remains controversial. And it's very scary to many people. The World Health Organization and the US Centers for Disease Control had maintained that you have to worry only about 2 types of transmission. That from inhaling respiratory droplets from an infected person in your immediate vicinity, or less common, touching the surface of a contaminated surface and then your eyes, nose, or mouth. The WHO officials did acknowledge that the virus can be transmitted through aerosols, but say that only occurred during medical procedures such as intubation, where there's a large number of particles emitted. They claimed, the WHO officials claimed that the theoretical arguments based on lab studies or modeling rather than evidence from the field was not sufficiently convincing to consider airborne transmission as having an important role in the COVID-19 spread. But when scientists say there is no evidence, sometimes that's because the evidence is genuinely hard to collect. Let's first take a look at some of the anecdotal evidence that existed in the early days of the pandemic. You're probably familiar with some of these classic examples where there were significant outbreaks that took place on cruise ships, restaurants, and a church choir practice. Many outbreaks have occurred in prisons, bars, meatpacking plants, and weddings, anywhere that people congregate unmasked for an extended period of time, mainly indoors where there's poor ventilation. This story has been repeated over and over again, and yet government officials feel pressure to reopen shuttered businesses well before it is safe to do so. Early studies in Singapore, China, and the US did find the coronavirus in air samples and on swab samples of the air vents and floors far away from the infected people. But they had not yet seen any publications with viable virus found on any of those air samples. So while the presence of the virus suggested that it could be transmitted by air, there was still no direct evidence of airborne virus infectivity. That changed in August, when 2 studies were successful in sampling infectious airborne SARS-CoV-2 virus. Dr. C.Y. Wu's group at the University of Florida was the first to report viable airborne virus in a hospital with infected patients at a distance of up to 4.8 meters. This was widely reported by the media as the smoking gun, proving that the virus was viable as an aerosol and at distances far greater than the 2 meters we had been told was the unsafe distance. Airborne transmission was not only possible, it was highly probable given all of the other evidence that existed. but the World Health authorities were slow to recognize this new information. In response to a letter that was signed by 239 scientists urging WHO to consider aerosol transmission, WHO made an addition to their website on July 9th, recognizing that airborne pathways may play a role, but that more studies are urgently needed. The US CDC was still reluctant to make similar statements. But then, more than 3 months later, on October 28th, the CDC finally admitted that COVID-19 can sometimes be spread by airborne transmission and likened it to the way that tuberculosis, measles, and chickenpox spread, and that it can happen at distances more than 2 meters away. The fact is that bioaerosols can be very challenging to sample. Traditional sampling methods like filtration, impingement, or impaction are relatively old technologies and are generally inexpensive. The problem is they also don't work very well. Some of the most serious deficiencies are the size-dependent collection efficiency, the dilute samples making detection more difficult, and there's a potential for contamination. Because of all the deficiencies in bioaerosol sampling, there are no regulatory standard methods for sampling them. Very often, the sampling methods themselves damage the microbes and can inactivate the very viruses and microorganisms you're trying to evaluate for infectivity. These stresses include desiccation, mechanical stresses, or thermal shock. So our new approach for sampling bioaerosols uses a method of condensing water vapor onto the small particles to form a fog of microdroplets. The water blanket that surrounds each bioparticle provides a hospitable environment and allows gentle, stress-free sample collection that interfaces with a wide variety of analytical approaches. Our technical advantage is the uniform, high-quality sample with low contamination. The gentle water-cushioned collection keeps the cells intact and maintains viability. And by sampling directly into a genomic stabilizer, we can instantly preserve the DNA or RNA for the highest genomic recovery. The stabilizer inactivates the microorganism or virus so that it is no longer infectious, making it safe for handling and an ideal sample for molecular analyses like PCR and sequencing. The photos shown here are of 2 different research-grade bioaerosol samplers named the Spot Sampler and the BioSpot-VIVAS. So laminar flow thermally diffusive condensation particle growth was introduced in 2005 by Dr. Suzanne Herring and was first applied to condensation particle counting. This was used to measure the number concentration of ultrafine particles. It relied on passing aerosol through a wet-walled growth tube that started out cold to condition the aerosol and then passed it through a heated section. In the heated initiator, water evaporates from the wet walls into the aerosol stream. The water vapor diffusivity is faster than the thermal conductivity, creating a condition of vapor supersaturation in the airstream. This unstable condition forces water vapor to condense onto seed particles as small as 5 nanometers and rapidly grow them into droplets that are supermicron in size. Then in 2014, a new design was introduced that added a 3rd temperature stage called the moderator. The moderator again cools the air, which continues droplet growth while simultaneously removing water vapor to the wet walls, reducing the dew point of the aerosol stream at the exit of the growth tube and avoiding unwanted condensation downstream of the growth tube. This 3rd stage made it practical to apply the laminar flow condensation growth technique to particle collection. The enlarged droplets are collected by impaction. On the left, we see an image of the growth tube in its normal operating orientation, a vertical orientation, with the aerosol sample inlet at the top. The droplets that are formed within the growth tube are passed through an impaction nozzle and are gently collected either into a liquid, as shown here on the left, or onto a solid surface. The charts in the middle show modeling calculations where we have temperature zones, 3 different temperature zones, cold, hot, cold, but the flow itself doesn't ever experience excessive temperatures. In this model, the temperatures never exceed 30 degrees C, which is very typical of the conditions in the human lung. The middle chart shows the supersaturation ratio that's formed within the initiator section, and levels of up to 140% activate condensation on the seed particles as small as 5 nanometers. The bottom chart shows the droplet growth, and you can see that it grows to a nominal 3 microns in this particular model, which makes it very easy to capture them with bounce-free impaction. And the cushioning effect of the droplets protect the cells so that they remain intact, and when sampling into liquids can remain viable. Because the physical size of the particles are enlarged, we get uniform high collection efficiency over all the sizes of interest, including the very small viruses, toxins, proteins, and metabolites, irrespective of the bioparticle shape, material composition, or hydrophobicity. This collection efficiency is then compared to the very commonly used, the standard biosampler impinger, which has been used for many years, and also the high flow wet walled cyclone, which can sample at hundreds of liters per minute, but you can see that the collection efficiency is very dependent on particle size. So how well does this condensation growth capture technique work for bioaerosols? Here I'm going to borrow some images from Dr. Wu's work and his team at the University of Florida, where they were using early prototype condensation growth samplers. This was a collection efficiency of laboratory generated MS2 particles, which are about 30 nanometers in size, and comparing that to the SKC biosampler, the impinger. You can see that in as far as collection efficiency goes, the capture efficiency for the condensation growth tube is about 10 to 100 times more efficient. Here's a chart showing from the same group showing the collection of laboratory generated viable influenza virus particles. You can see again that the condensation growth tube sampler has a much higher collection efficiency, but also recovery of the viable virus as compared to the SKC biosampler. Now we only get about a 10 times, 1 order of magnitude increase in capture efficiency and recovery, but this is because the influenza particles are larger in size, typically around 100 nanometers in size. And so we would expect that the impinger would be somewhat more effective. But the main thing is also that the recovery of the viable aerosol virus is also enhanced. In another study showing capture into a genomic preservative, this is a study done by the University of Colorado in Boulder. And on the left side, we see capture using the condensation growth tube technique, capture onto a solid surface or into a liquid. In both cases, the solid was pre-treated with a coupon, pre-treated with the genomic preservative, and the liquid was directly sampling into the genomic preservative. And we don't really see a statistical difference between solid capture and liquid capture when sampling into the genomic preservative. The chart on the right is really a result of a question that we had that, well, if sampling into this genomic preservative is such a wonderful thing, why do we need the growth tube? Why don't we just impact directly, you know, these bacteria and fungal spores and the, you know, the larger sizes that are easy to capture? Just impact them directly in the preservative. Why do this growth, droplet growth? Well, we did that test. And so we used the growth tube and not using the growth tube. And we do see a statistical difference in the genomic recovery between solid and liquid, or between the growth tube and not having the growth tube. And so it does show that by forming the droplets around the particles first, we get enhanced recovery. These are just some of our early adopters. Prior to the pandemic, they were looking at disease transmission routes and were sampling pathogens in environments as diverse as a swine barn and a hospital operating suite. But many, or I should say most of these users are now sampling the SARS-CoV-2 in a variety of settings. So thank you very much for your attention, and I want to give a big thank you to our funding supporters as well.
Thank you very much, Pat, for your very interesting presentation and for this focus on sampling systems, in particular on the condensation growth tube techniques, which, as you showed, gave some important results in the improvement of bioaerosol sampling. So thank you again. I don't see questions at the moment, so we would have time to answer some questions, but maybe we will receive later on. And so if there are no questions or comments, we can go on with the following speaker, Professor Pierluigi Barbieri. So Professor Pierluigi Barbieri, together with Dottoressa Sabina Lichen from the University of Trieste. Professor Pierluigi Barbieri is Associated Professor of Environmental Chemistry at the Department of Chemical and Pharmaceutical Sciences of the University of Trieste. And he is active in the multidisciplinary initiative of the University of Trieste for the development of technologies for the contrast of the spread of SARS-CoV-2. So thank you, Pierluigi, to start your speech. The title is Challenge for Bioaerosol Research from COVID-19 Pandemic and Interdisciplinary Approaches. Thank you, Pierluigi.
Thank you to the organization that I'm involved with. I share. Can you see the presentation now? Okay. Sono in condivisione.
Ecco, nuova condivisione.
Can you see the presentation?
Yes, I can. It's okay.
Thank you.
You're welcome.
So, just okay. Thank you to the organization for allowing me to share some thoughts with you. My speech is not strictly scientific report on some data we collected, but it is an introduction to the relevance of the issue we are facing now, based on say literature research and some proposals that we pose to the scientific community and technical community. Many things has been already introduced by Pat Kitty. We are talking about, say, the presence of virus SARS-CoV-2 in the aerosols, and in March and April, indeed, 2020, some very relevant publication were, say, released showing how SARS-CoV-2 can stay viable in controlled situation in the aerosols, pointing the interest to the airborne transmission of COVID-19. Immediately after, we had some WHO strong statement stating that COVID-19 is not airborne. This moved somehow the researchers to deepen the knowledge and search for the presence of the virus in the aerosol. This is kind of small, say, data that show how in 2020 there is an increased number of paper with bioaerosol as a keyword, because significantly higher than previous year, and we are starting also in the 2021 already with a good number. Many things we know since before 2020, so focusing on the relevance of close contact in the transmission, and then the deposition of say, particles that are emitted by infected people. So the possibility of transmission of COVID disease by fomites. The simplification of focusing of particles smaller than 5 microns and as aerosols, and particles bigger than 5 microns is indeed a oversimplification that has been highlighted by many aerosols researchers. Here I cite simply Don Milton, that has published very clear results on how also in the short range it is not say droplets with a dimension higher, bigger than 5 microns playing a role, but also the say bioaerosols with a smaller dimension that plays a role. It is well known from physiology that we emit particles that can say carry the virus with in a spread of in very wide interval of dimension, and it is depending on where these small droplets are formed, either from bronchiolar area with the smallest particles, and then in the oral cavity. And as it's told before, the aerosols are relevant in the short range, not only droplets. This is a very relevant implication also for what concerns personal protective equipment. So we do not need to care only for, say, face mask blocking just coarse air particles. There is a very fastly evolving knowledge on transmission, as was cited by Professor Stenner in the beginning, on first days of February, Nature published that again that coronavirus is in the air. So that we need really to focus research on characterization of the presence of virus in the aerosols. There is growing literature production on the presence of SARS-CoV in the aerosols, searching presence of RNA of the virus, both in public areas or public transport, have been clearly found, but and also in the healthcare area. It is why the say problem, the one of workplaces, and beside the trace of viral RNA, what remains open is what is addressed indeed by the condensation growth tubes, so the viability of the virus present, of the virus in the aerosols. The approaches are several for sampling. It is interesting that beside aerosol scientists, virologists starts to support the airborne transmission, even if they say, okay, we should understand how much it is relevant in comparison of other way of transmissions. I pose to your attention some aspects that to me as an environmental scientist dealing with air quality emerge very neatly. So if you go to examine the literature and you say you focus on a type of sampler and sampling length and flow rates of different studies, you observe very diverse devices and sampling time and sampling flows. So the volume of air sampled is rather variable, with devices that, as brilliantly reviewed by Professor Wood, that we'll talk in later during this symposium, has some strong point and a weak point. Impactor and cyclones, impingers, filters, electrostatic precipitators, has many drawbacks, and when compared to water-based condensation devices. In particular, as Pat Kitty showed before in detail, the efficiency for collection of small virus particles is low for impactors, impingers, and electrostatic precipitators. What I do point at your attention is that sampling time and flow usually are very diverse in literature and often rather small. So since the presence and concentration of virus in the air is not constant due to variable emission from patients and say the settling time of the particles depending on the size and dispersion in the environment, sampling for short time can be a problem. This issue of say, need of taking care of the variability of emission sources is pointed also by Professor Jimenez, that show how, for instance, there are super spreaders person that can contribute, and so going to sample for short time can be a problem. What is highly pointed by Professor Jimenez is that it is difficult to sample infectious virus, but this point of problematic sampling is shared also by other type of virus. So the VIVAS sampler is welcome as a significant technological advance that will be, I think, detailed by Professor Wu. What is highly needed now to identify the different potentially critical situation to focus on. So to somehow identify procedures that can be useful for characterizing healthcare unit characterized by low risk setting or other potentially critical situation as crowded areas, for instance. For each of the different situations, understand which are the best suited technologies and procedures, and look for consensus harmonization on sampling and analysis. I am very fond of a long-term monitoring to understand, to have under control, say, potential sources or critical situation. For this aspect, we propose usually a two-phase or two-stage approach. One is very rough, in which we collect on filters, say, particulate matter, and then we go for extraction of genetic material and identification by real-time quantitative PCR. If this kind of screening on presence of RNA show that the cycle threshold is low, in which appear positive results, so pointing at a high number of copies, then you can go for sampling gently in order to keep virus viability and proceed with cytopathic assays. Sorry, it is written badly here. Till now, we consider multi-stage impactors with gelatin filters or biosamplers or cyclonic devices. But what we are eager to know better is say, which are the results of sampling with the water vapor growth that is proposed by the technologies, which is one of the focus of today. Okay, our research group was successful in finding, say, viral RNA from SARS-CoV-2 with this rough approach of phase 2, both indoor and outdoor, and that's how I do finish my presentation. Thanking my coworkers and you for your attention.
Thank you very much, Pierluigi, for your interesting presentation and also to be on time. Thank you for your focus and also to have stressed the need to show common methods in order to reach a consensus on methods, on experimental approach to follow in order to obtain good results in experimental studies on bioaerosols. So, of course, this is a very important point, a key point. We don't see specific questions. So please, if you want to do question, use the D and R function. For example, before we had a question for Pat Kitty, and it was done by using the chat. So now, as we have time, we can, in the meanwhile, answer the previous question. So, okay, sorry, there is a question. Maybe, Pierluigi, you can see the question. If you want, you can answer the question by Alberto.
Which
I
problem
see
did
a
you
question
find?
by Andrea Gambaro. And also a question by Maurizio Gualtieri. Can you see the questions?
May I, do you hear me? What is
okay, yes.
What is our strong point is the collaboration with environmental genetics, being particularly good in extraction of DNA from complex environmental matrices. I would say that our first publication were done without specific say care, but they were referred to very peculiar environmental situation in which there was no awareness of the problem, so the spread of of the virus. Detail on detection time for the different techniques. You mean about sampling? So the preservative were okay. I can provide by email this this information. I have no perfect. Maybe Professor Provella later can give some detail on the issue.
Thanks, Gigi. I will give not the detail, but a presentation upon the limits of detection, even if not in environmental samples, but translation from medicine to possibilities for the environmental detection by stressing the concentration and the time of infection. So I'll talk about that in the very my presentation in, I think, 15 minutes after the other presentation upcoming. Thank you.
Okay.
Pierluigi, I see that there is also a question under the chat. So, and also there is okay, we have 4 questions under Q&R and 1 question under the chat. I don't know if you prefer to go on answering orally, or you can also answer in the writing form.
Oh, I'm not able to.
Can you see the questions?
Yeah, about universal target sequence for several viruses. I will let my microbiological colleagues to answer to this.
Okay.
No, there are no specific general target sequences for viruses. General at the same level of 16S, 18S, or ITS. So there are conserved proteins, of course we know, but it's a super, it's a harder, yeah, unfortunately.
Just to recall, sorry to interrupt you, that in human diagnosis in a diagnostic lab, most of the sequences of coronavirus should be evaluated, not just one, in order to be sure to correctly and properly detect the infection. So most of the protein, nucleocapsid, E protein are involved in those detection protocol. So the golden standard, well, we follow the path traced by CDC, but anyway, we have to work on most of the encoded regions of SARS-CoV-2. It's not as easy as for bacteria, I mean.
Okay. Pierluigi, I think that you answered all the questions, or maybe Professor Crovella will answer some of these questions during his presentation. So if you want to add something, or if you prefer maybe answer in the writing form to,
of course,
the...
One of the points is to extend experience we had for some aspect. We had for outdoor sampling, we had very peculiar, say, event, which is not easily reproducible, and we need to say extend, say, experience on a monitoring campaign. It is much easier in indoor situation, and this is where we are focusing by now, our research group in healthcare units, which can provide, say, validation somehow for, say, more controlled situation that we have. Starting from that, we plan to extend to relevant outdoor condition. But our gym, say, now it is in indoor, say, settings.
So there was an earlier question about the genomic preservative used with the condensation growth capture, and I just wanted to let you know that the data that was shown was using a product by Zymo Research called DNA RNA Shield, and we'd be happy to give more detailed information about that, but that's what we had used for that experimental test.
Okay. So I let the word to my colleague Francesca Malfatti.
Yes, so it's a pleasure to introduce Professoressa Francesca Malfatti from the University of Trieste, and also collaborating with the CASPO, CIO in the USA. She will give a talk on marine microbes structuring ocean atmosphere ecosystem. And I give the word to Francesca, and thank you for your presentation, Francesca.
Hi, happy to be here. It's a great pleasure to talk with such a broad audience spread all over the world, new friends and old friends. From the pamphlet, you have seen that I got a fish, so for sure I will not talk about COVID-19, because I was in Italy when everything started, and I didn't focus on aerosol, but I focused on sewage. So I didn't want to talk about that. I would like instead to tell you a story that I have been working with a case that is a NSF NSF Center for Chemical Innovation that has been established in 2014 in USA, and we care about how sea spray aerosol can influence the chemistry of the environment, the chemistry of the atmosphere and the climate. So if you wish really a big, grand goal. I am a microbiologist and I like to study bacteria. Let me know, actually, I need to ask you a question. Can you see my screen? Okay, thanks. I would like to go full screen if I manage. Control L. Now I see nothing. Oh, sorry. Luigi. Sorry, I have a black screen, but I will not go full screen. Let's go. So today we will explore marine bacteria and also sea spray aerosol bacteria once they are ejected from the ocean into the atmosphere. So let me introduce a little, I will give you a little primer about the microbes. Then we will see how ocean and the chemistry of the ocean and the biology of the ocean are structured by the microbes. Then we will follow into how microbes structures atmosphere, chemistry and climate, and then I would like to wrap it all up. Thank you. I am a microscale microbiologist, so I put bacteria in their context. They are interacting with molecules one at a time. They are living in a small world, a microscale world. In this picture I took at the laser scanning confocal, you see a 0.5 nanoliter of seawater, and the green dots are marine bacteria that are clustering around some hotspot of organic matter. And you see some orangish things that are the chloroplast of microalgae that are living in the ocean. So I like to study them and interrogate them at the real scale in the real environment in order to avoid artifacts and put this in a big ecosystem context. What are microbes doing in the environment? Well, they were here first on planet Earth. Because of their biochemistry, they have structured the planet, and also they are keeping each ecosystem functioning. Us, our dog, our parrot, the ocean, the soil, and the atmosphere. So they are really highly abundant and very diverse. They have a great range of metabolic capabilities that we call also biochemistry. And they are keeping the ecosystem functioning because everything that dies on planet Earth will be possibly recycled by microbes that will then allow the nutrient to flux through other biota. And so life will start again. Of course, microbes, while are degrading organic matter, they are respiring, produces gases, and they are transforming the matter from organic to inorganic. And so just again to tell you, I like to be in this cozy environment where bacteria and archaea are interacting with viruses, or they are betrayed by protists. They are releasing organic matter, producing CO2 and or producing oxygen if they are cyanobacteria. When we talk about biotransformation, that I know it's an edgy definition, we are referring to the enzymatic activity bacteria do while they are remineralizing the organic matter and degrading it. And they don't see, they can move, but they smell. So their enzymes, that is the digestive gut, like the enzymes we have in our saliva, are arranged on their surface. And so while touching and interacting, they are degrading the organic matter and changing their microenvironment. And so it's a very intimate relationship. And because of this, the ecosystem is still functioning. And we don't smell sulfur or putrescine or cadaverine in the environment, because bacteria, the good guys, are constantly remineralizing the organic matter. Sorry for this prime. I know most of you are scholars, so take it easy. So what about atmospheric structuring? Well, ocean and atmosphere are just two spheres, one more viscous, the other not so much, and they are constantly interacting to each other. We focus at CASE on sea spray aerosol that are formed by wave breaking and bubble bursting, so microscopic particle parcel of the ocean are launched in the atmosphere. And the marine aerosol have important properties such as cloud condensation nuclei or ice nucleation nuclei, or for forming clouds, for instance, and then precipitation, but also they are important for absorbing or not the solar radiation. So there is a great amount of sea spray that is ejected. And now if you think about microbes, and in a liter of seawater, you have heterotrophic bacteria, I will focus mainly, between 10 to the 8 to 10 to the 9 cells in a liter. Ocean and atmosphere are constantly talking. They are exchanging gases both ways, and also microbes, and also chemicals. And so then, if we have a look at the atmosphere, we have a wider range, because the atmosphere is a little bit tricky, and also there are many layers in the atmospheres that are always challenging to samples. So we have 10 to the 2 to 10 to the 4 bacteria cells in a liter of sea spray aerosols. So what are they doing? If they are living and if they are constantly degrading, what is our framework for asking questions about the role of bacteria in structuring atmosphere? So we know that if they are doing it in the ocean, why not? Can they doing it when they are launched by film drops and jet drops? And then how do they control the gas release? So within CASE, we have been focusing to targeting biotransformation. I am a microbiologist, so simply I started asking the question, can I detect enzymes in the sea spray aerosol? It took a while, but finally I was able to publish in 2009 the discovery that in sea spray aerosol collected at that time with an impinger, now we are using the spot sampler, I was successfully able to measure proteases activity, lipase activity, alkaline phosphatase activity, glucosidase and chitinase activities. So these are all the enzymatic repertoire that bacteria uses to cleave the organic matter, to remineralize what we call the dead things or organic matter, and then to release the nutrients to our biota. So this was a transformative discovery, because from, you know, an atmospheric chemical and point of view, who cares about biology? You know, okay, biology happens, then they die. Instead, it's the chemistry driven, the thing in the atmosphere. Yes, but also with the help of biology. So within CASE in 2004, using our 30-meter long wave tank that is 13,000 liters, we were able to create a plankton bloom, so microalgae bloom. So we give some nutrients and the microalgae were really happy. And then we were measuring again enzymatic activity in the water, and we saw that there were two kinds of lipase activity that somehow has shaped the organic enrichment of the sea spray aerosol, they measured by AMS. So a mass spec, a very simple mass spec. And so what we saw was that because bacteria were actively degrading the organic matter, the lipids in the ocean, in the water, we were seeing a feature, a signature that was driven by the chemical activity of the bacteria in the water and was transferred in the sea spray. We were very excited. We also asked the question, which are the guys that are doing, and can we see them in the SSA? And so we did a little bit of metagenomics, and then we did the 16S, not 18S. I know, let's not talk about it, but that is what we did. And so then we saw the fingerprint of the bacteria that were ejecting, and we have postulated the idea that the surface and the genome features were dictating the transfer. Still, we are investigating this. Now, when you think about that, you are breathing a clean ocean versus you are breathing the sea spray, the spray that comes up from a sewage plant, that has a health implication, of course, because we know many things can be transferred by the aerosol and still are pathogenic or virulent to us. In 2018, after BEAST, we set up an experiment that we call BEAST, where we were focusing about sulfur transfer. So we were always growing phytoplankton and then moving to a marine aerosol reference tank, MARC, that we use. It's 110 liters that we use to generate the real continuum of shape of the sea spray aerosol, because chemists like Gigi tells me that only if you have the right size, you have the right discrimination and ejection of chemical moieties. That being said, I was interested in challenging my bacteria, and so I use a free bacterial strain that I knew and I work with, and I know the genome, that I call a mock community, because I wanted to challenge the ocean bacteria, the water bacteria, in order then to predict SSA change in terms of chemistry and the selectivity transfer. The experiment is still, and this is the first time I have personally used the spot. So we had to target, of course, the collation time and the flow rate. And so that is, part of the art of doing science. So when we add the bacteria isolate, then the two community refers a circle as a mark that was not amended, mark A, and square mark C that receive the isolate, then they diverge over time. So in a way, yes, great. I was able to mess around the community and mess around the interaction between the microbes at the macro scale, at their scale, and also we were seeing different features and different bacteria being ejected, and we are still analyzing the SSA data. Following on the discovery of the enzymes, we wanted to see if the enzymes, once they are ejected in the sea spray aerosol, have the potential to change a chemical shift measured by ATOF-MS, aerosol time of flight mass spec. And so then we created a bloom. We let it flow through a reaction tube where we have mixed some nitrate, and then we have measured afterwards what was the chemical feature, and also we have measured the reactivity. And so what we found, and Chris Lee and I, we are still finalizing the writing, we saw that the reactivity was decreased upon the protease amendment. So because of we have a molecular model in our mind, we see that the protease changes the solubility of the casein, that is the protein that was hydrolyzed, that protecting more the SSA particle, so decreasing the possibility to react with NOx species. And that has potential consequences for the reaction that we see are happening right now in our atmosphere. And the last thing is 2019, the last big experiment we did pre-COVID, where we have stimulated 3 different blooms. So we created a different trophic scenarios with more and more and more microalgae in our super long wave flume, 30 meters long. And now again with the spot sample simpler. I have coupled the excitation and emission matrix analysis that is telling us something about the biological feature based on fluorescent categories that have been chosen, such as chlorophyll, tryptophan, and other amino acids. And so I was able to then I was able to see the bacteria that were metabolically active in the sea spray here in gray and in the seawater here in blue, based on the spot sampling collection that were matching also the profile that I was getting from the excitation and emission matrices that you can consider a bulk measurement. And so this was important because we would like to know, once the bacteria are ejected in the sea spray aerosol, what are they doing? How fast are they growing? And are they respiring? And so far, you know, it's difficult to go in a cloud chamber. I know there is one in Caltech, and I know that in Genova there is a fantastic chamber, but it's not a walking chamber. So for me to be an ecologist, microbiologist, to have the microspatial context within the aerosol is the dream of my life. Consequently, I'm trying to use different methodologies in order to ask these simple questions. Once they are ejected from the ocean into the sea spray, what are they doing and how are they doing? With that said, I hope I have conveyed you this excitement that we have in case, and I have, and I hope also to start working COVID after the COVID with Gigi even more tightly and with my other colleagues at the Department of Life Science to really give a push in sea spray and microbiology and their impact on the atmosphere. With this, I would like to acknowledge, of course, Università di Trieste, where I'm at, the CASE, the center that I am part of, and the NSF for funding. Thank you for your attention.
Thank you, Francesca, for your speech and for this focus on marine bioaerosols and for sharing your research results on this so relevant topic. And I think that there is a question that maybe you want to answer by the same attendee who did a question before, and you answered about I hope that you can see the question.
Spettatore anonimo
yes.
at 4:22 p.m.
Yes.
So, okay, so a person said, so it is not possible to identify which viruses are present in the environment easily together at the same time, but there are any other parameters that can help identifying viruses present in the environment? Well, let's consider that we have been to the moon and we go to Mars. So if we now can answer this question considering these two achievements, I would just like to say, if you have the right sampling device and then you collect enough biomass, you can do metagenomic, shotgun metagenomics. That is difficult, but there are virologists who are really expert in dealing with it. I mean, not all kind of science can be done with a spoon and a candle. You need sometimes a very expensive machine. So this is what I think, because I not always use the Petri dish and the Bunsen burner. I do also something a little bit more complicated, like atomic force microscopy. So you see, I would say there is a hope, but you need to have a vision and the investment, and also the desire, the urgency. As you have seen that within the COVID-19 pandemic, everybody is doing what they can to solve it. Allora, Velosi was already. Andrea Gambaro said, what do you think about the problem of storage of samples before analysis? I think you answered it. Gualtieri, given the limitation of the filter samplings, which are the problems with your quartz sampling, and what about your, no, maybe about the extraction of RNA? Allora, maybe Gigi already said. Can you give the type of detection limited different things? I think I don't have any.
No, I think we don't have other questions.
That's great.
Thank you, Francesca. So thank you all speakers, because we are perfectly on time, and so we can go on with the speech by Professor Sergio Crovella. Professor Sergio Crovella is a full professor of biology and genetics at the Department of Biological and Environmental Sciences at the College of Arts and Sciences in the Qatar University, Doha State of Qatar. He has a PhD in molecular anthropology, and his main research interests are on host response to infection, autoinflammatory and autoimmune disease, as well as integrated omics analysis. So thank you, Professor Crovella, for your speech. The title is Bioaerosol Analysis, Development of Procedures for Sample Preparation, RNA Analysis, and Infectivity. Thank you.
Thank you very much, everybody, the audience and the organizers, for giving me the possibility to share with you our quite simple and rough analysis and our experience derived from clinical samples, trying to apply this knowledge and to give a hand and to help our friends working on the environment. The presentation is made by me, but I wish to thank Luisa. Luisa Zupin is the senior postdoc who was pivotal in contributing to do any kind of experiment dealing with the virus. So let's go rapidly to the matter of debate. The question is, and it's a question still matter of debate, how, by which means airborne transmission of SARS-CoV-2 occurs, and if there is a true airborne driven transmission that could be replicated not only in wonderful publications, but also in the routine practice. This is a topic that we are developing together with the equipe of Gigi Barbieri at Trieste, trying to find out some easy, possibly inexpensive ways to get information about the potential infectivity of the, let me say, genetic material of the virus present in the air, sampled in the environment. Everybody of you know the difference between respiratory droplets and aerosol droplets, so I'll not go through this aspect. One is bigger, the other is smaller, and the velocity of travel is, of course, quite different. Let me first recall a very important topic. Most of the studies on environmental samples have been done by just picking up, recovering an RNA signal from the virus, many times just getting partial information on a small portion of the virus. So the pitfall, technical pitfall, was how to identify an eventual trace or presence of an intact viron, and is this viron able to infect us, human beings. To answer this question, no way. I mean, you have to perform a test of infectivity on at least a cell model in a BL3 laboratory. So what we did trying to contribute with our forces, which are coming, I repeat, from laboratory medicine, was the evaluation of a possible threshold cycle related to infectivity, working on samples derived from patients, working on serial dilutions of those samples in order to find out this threshold. Which is the threshold? This is not fully applicable to environmental samples, but which is the threshold that is useful for diagnosis? I mean, we don't use in-house methods to detect RNA. We use robust kits validated and stamped by the CDC or by health agencies to detect the presence of the virus. So we wanted to observe which is the minimum threshold cycle able to produce a cytopathic effect by SARS-CoV-2 on an in vitro cell model. The in vitro cell model is very simple, is widely accepted and easy to be cultivated, is Verocell line derived from renal epithelium of Cercopithecus aethiops monkey. The experimental design is really trivial. I mean, we take swabs from COVID-19 patients and we test the presence of a cytopathic effects and the presence in the supernatant of replicated virus after up to 7 days. So we see in time, during time, which is the fate of the virus. So we respond to the first question, which is the threshold cycle that gives us an idea of the possible infection, and overall, do after the infection the virus successfully replicate? Can we find the virus in the supernatant? Do the virus is able to kill the cells? So very simple questions that we wanted to respond. First of all, we interrogated several samples. Here you have just partial numbers that we obtained, and we have seen that we have successful infection with small CT threshold cycle. So this is not a novelty. I mean, everybody all over the world found this result. I mean, the much virus you have, the greater is the possibility of infecting your cells, even if we had some failures. But the interesting finding in our experimental setting was that even at high threshold cycle, the virus was able to infect, to survive, to replicate, and we have been able to find it out after 7 days. So giving us a first warning about the small quantity of the virus. This result, even if is not so original, but give us or give to people managing the health policies the possible amount of virus, which is very little, measured is in inversional proportional to the threshold cycle. So a minimum quantity can be effective in infecting. So just to show you, we calculate after 7 days in the supernatant the quantity of the virus, as well as we observe the cytopathic effects here shown. Those are the not treated cells, and those are the treated cells who are showing a clear cytopathic effect due to SARS-CoV-2, not only infection by replication in vitro. So our first set of results showed us that the successful concentration has a threshold cycle of 24 CT every time, everywhere, in a very simple experimental setting. However, even if we have initial viral load very poor, we can have disinfection. we can have a replication. So this is a warning. I mean, we cannot say, okay, guys, when you have something above the 24 CT, you can stay quiet and safe. We have seen that 35 CT corresponding to E elevated 0.4 viral copies is still able to infect, not only to replicate and to produce a cytopathic effect. So after this, we wanted to interrogate our very simple system, trying to find out, trying to focus to the time of infection, the so-called lift time. If we spend some time in a lift or in a very close environment with other people, which is the possible time of exposure able to cause an infection. So same model, we applied our model using an MIO, number of virons added per cell during infection, same MOI, different times, 5, 15, 30, 60 minutes, and different concentration going from bigger to ridiculous concentration of the virus, and we tested our system. This is quite impressive. I mean, just after 5 minutes of infection, and we are testing now 1 minute of infection, the virus is able, after 1 week, to successfully replicate and damage the cells. So 5 minutes of infection, enough for the virus to successfully infect and replicate. Then MOI, quantity of the virus. I mean, with a quite small MOI, 0.01, around 100 virions per cell, we have an infection, we have a cytopathic effect. Here you can see that at 5 minutes of exposure, with different magnification, you have a cytopathic effect. Same thing for the different MOI. Here at 0.01, you have cytopathic effect. Another technique, much more easier and not expensive, the crystal violet staining, show that you have a cytopathic effect for all the time of exposure. And here, with the MOI 0.01, you have the limit of the number of copies able to infect human cells. So, the cytopathic effect was evident at any time, but we focus our attention at the 5 minutes, which is a lift which is going, it's an Italian lift of the very old houses, but 5 minutes, it's a very small time. And also the quantity is quite small. 1,000 infectious virions are able to cause the infection. We are going down the five minutes now. Then we interrogated ourselves about the limitations of the technology employed, and we decided to change our strategy. The VeroCell line is the widely accepted in vitro model for employing a permissive cell to be infected by SARS-CoV-2. But we tried another model, a human one, the CACO2 cells. Of course, this model has limitation since those are immortalized cell lines derived from colorectal adenocarcinoma, but at least they were humans and not monkey-derived. Same experimental design, same times, same MOI. Here as well, we have seen that in human cell lines, again, 5 minutes of exposure were able to increase after 7 days the number of viral copies. Same for the MOI. We obtained very similar results to what we obtained on VeroCells. So, given quite an important warning for this for humans, not for the air, but an important warning about social distancing, and at least a warning. I mean, we didn't want to scare anyone, but a warning about the capacity of this virus to infect rapidly and with very low quantities, which was something quite expected about the clinical data of the pandemic. The interesting thing is that in this immortalized cell model, we haven't found any cytopathic effect. However, the virus was able to replicate, but cells were not dying, at least after 7 days. We can figure out if those cells will survive after 7 days. Of course, they are immortalized cell lines, so much more strength with replicative fitness higher than normal cells, primary cells. Same for Demoy, no cytopathic effect. So we have been curious, and this is a very nice slide that Louisa provided for me. We searched by using an antibody for the SARS-CoV-2 nucleocapsid, the presence of the virus in the cells, and you can easily see that the cells are full of virus. In blue is the nucleus, and in red we have the F acting. So much more robust demonstration that the virus succeeded in entering those cells. So what to say? Our findings very simple. Just say, okay, guys, a warning for maintaining social distancing, for saying that the virus is very effective in small quantity and in very short time. And we do hope to receive from Gigi the samples from the air sampler to test the environmental samples recovered in the hospital of the COVID hospital of Trieste in our very simple experimental set. So, of course, a closed environment set and poor air circulation, it is obvious, but should be considered when working with the health policies. This is just a small publicity propaganda that we are doing by studying the literature and not so happy. It's fantastic, the Lit COVID, the NCBI tool for studying the literature. We designed another platform called the Plat COVID, able to not only retrieve publications specific for this is our bioinformatics soul, not only specific for the literature, but also allowing scientists to curate and comment the literature. So I do thank you all, and really a special thank to Tecora, who supported us for this adventure. We started from nothing, Louisa, me, and two persons who have been key persons in this research, Dr. Francesco Fontana, microbiologist at Monfalcone Hospital, and the head of the laboratories of the Friuli Venezia Giulia region, Dr. Maurizio Ruscio. Thanks to them, we have been able to apply those models in a proper BLS3 laboratory. Thank you very much again for your attention.
Thank you very much, Professor. And I see that we have two questions by Giovanni Franco Crosta. One is for you, Professor Crovella. Can you see the question?
Very good question. Thank you very much, Giovanni.
Maybe you can read the question because the other attendees can see.
He's asking if there is a significant variability in RNA of virus strain used in our experiments. I mean, this is a very good and interesting question. I can say that we are working, now sequencing the virus is quite easy. We don't need to possess an NGS instrument at home. We are working now with the British variant, and let me say that we are achieving the same results in our cell models of the previous variants. So up to now, but we have a simple cell model, is not so affected by the viral strain. Of course, this is a topic of great interest. We are receiving samples from the clinics, and if the clinics are able to identify the virus, or if we can sequence the virus, as we did for the British variant, we can compare the different strain. Up to now, we have no difference between the strains. I mean, of course, this question arises upon depending upon a query, not only in the scientific literature, but also coming from the bombing of information. One variant, we have no Brazilian variant, and we have not yet South African variants to be tested. Are they more effective or not? I mean, up to now, our model just demonstrated that the British and the circulating variant in Italy had the same pattern of widespreading, I repeat, in a very simple cell model. Satisfied, Giovanni?
Okay. Yes, okay. I see that we have other two questions. The other one is always by Giovanni Franco Crosta for Francesca, and then we have a question on the chat. But just to be on time, I would like to go on with the last speaker, and then we can end with the pending questions at the end. So we respect the table time if the speakers agree.
Thank you very much.
Thank you, Professor. And Tanya, if you want to introduce Professor Wu, our last speaker.
Actually, Tanya's asked me, and I am very pleased to be able to introduce Dr. Wu. Dr. Chang-Yu Wu is a professor at the Department of Environmental Engineering Sciences at the University of Florida. And he received his bachelor's degree in mechanical engineering at National Taiwan University and his master's and PhD from the University of Cincinnati. His research has ranged from air pollution control and basic aerosol science, environmental nanotechnology, dust control, and now more so in bioaerosol sampling. He has published more than 150 refereed journal articles and is a frequent speaker, invited lecturer. His research has resulted in 6 patents and 3 pending applications. He is a past editor of the Aerosol and Air Quality Journal and is an elected fellow from the American Association for Aerosol Research. And so, Dr. Wu, it is my great pleasure to allow you to give your talk. Thank you.
Thank you, Pat. Thank you very much. And I'm very honored to have this opportunity to share our research on the efficient collection of viable SARS-CoV-2 aerosol and using Pat's device. Okay. So let's move on to the next one. So everybody has seen this picture before. It has been commonly used and to represent the important modes of transmission. And this is from WHO, and because of the large droplet and the fomite transmission, and therefore there are recommendations in terms of social distancing and washing your hands. And as Dr. Barberia mentioned earlier, the aerosol transmission mode is ignored by the WHO before. So if we follow, the question is, if we follow the guidelines, and are we safe? Well, there are a lot of anecdotal cases. Also, this one mentioned earlier by Pat. So in the Washington State, Skagit County, and just the choir practice, 61 persons, among them 45 were infected and the 2 died. And they did follow the social distancing practice and they washed their hands and they didn't want to touch each other. But still, so many people got infected, and this is a perfect situation for transmission. And so that says, well, the WHO guideline is not really good enough, and why? And at the same time, they are happening a lot, and I'm just showing 2 examples. A lot of samplings in hospitals in different environments to show, hey, we have been able to detect RNA of this virus in air samples. So this says the aerosol is important, but still WHO doesn't recognize that. So why is that? So we went back to WHO guidelines. So according to WHO, they said there are respiratory droplets and they are 5 to 10 micron and larger. And aerosol is defined for those less than 5 micron, and they call it a droplet nuclei or aerosol. And please follow me with the second paragraph. This is very important because they define aerosol transmission as a spread of infectious agent caused by the dissemination of droplet nuclei that remain infectious. So if you just detect it by RNA, they don't care. They don't want to consider that. They will not consider that. And when suspended in air over long distance and time, so if you capture the exhaled breath, it doesn't count because that's right out of the infected person. It doesn't count. And they do recognize, as pointed out earlier, that pipette that during the medical procedure, they call it the aerosol generating procedure. They acknowledge there is such an airborne transmission, but others, they don't acknowledge. So we set out to study to assess if we can collect viable SARS-CoV-2 and greater than 2 meters away from a known source that is not an aerosol generating procedure. Okay, so we use a Vivas or BioSpot. We actually use 2 in this study, and Pat has explained the principle of this device. So I'm going to save the time for that. But essentially, you have these very tiny particles that are amplified and then get collected. And also pointed out earlier, the collection media is very important. So over here, we try to preserve the viability. Instead of killing the bacteria, the virus to make it safe for handling, here we do want to preserve the viability because that's very important to our mission. And we place our samplers in this hospital room. And this is the BioSpot and the Vivas over here, a patient 1 and a patient 2. In this room, the air change per hour is 6, and our hospital use MERV 14 filter, which is having 80% efficiency for 0.3 micron. It is pretty good. And also they use a UVC irradiation, and they recycle 90% of the air and only takes in 10% of the outside air. So this is the condition of that room. This is pretty good in terms of the ventilation. But still, we were able to capture the virus over there. But we actually have to say we were lucky. So this is the workflow for the study. So the emission discharge of the virus from the patient collected by Vivas. And we do 2 types of analysis. First is just to detect the RNA if there is such a virus, okay, just for detection like everybody does. And if yes, then we inoculate the cells, the Vera E6 cell or LLC MK2 cells with the collected samples. And we observe the cytopathic effects in infected cell, like Professor Corvella mentioned earlier. And then if after several days, we would harvest the virus and then purify their RNA and do again the quantitative analysis for how much of that. And then we also do sequencing to identify the genomic sequence. Okay, so let me show you the results that we have. Okay, so this one over here, and this we have the 4 samples on that day. And here is the distance, the 2 meter or 4.8 meter from the patient. And in the afternoon, a patient actually was discharged. So we put 0 over here. But as you can see, we still detected the RNA over here. And this is pretty good concentration. And the CQ value actually is high. A lot of people say you cannot grow it, but we were able to do that. And we have a very good virologist at our university, John Lenke. So as explained earlier, once you detect it, then you inoculate the cells with the virus. And then you would observe them on different days and harvest them. And here, this is the original day 0 and 4 days and 7 days and 10 days. You can see the cytopassive effects very clearly. So after that, we harvest the viruses on different days. And then you can see this one is closest to patient 1, has the highest concentration. Even at 4 days, you were able to detect it. And with more days, then there are more multiplication of the viruses. So you can see higher concentration represented by the lower CQ values shown over here. And even for the this sample with the lowest virus concentration, and after 10 days, we were able to detect the presence of these and quantify the amount of the virus after 10 days. Okay, and then we do the estimate of the viable virus counts based on our calibration. And this is the first time reporting the viable virus count per liter of air. And of course, if it gets closer to the patient 1 and you have a higher concentration, and the further away you have a lower concentration, but we were able to quantify that. And after that, then we did the genomic sequencing of the samples, and we deposit the information in the GenBank. And this is our second environmental SARS-CoV-2 aerosol sample deposited in the GenBank. And for this one, because most of the others are the human specimen, this is the environmental sample. And the virus genomic sequence it agrees with the predominant virus lineage in the circulation in the USA as of July 10th. And very importantly, it's this one. So we also had the samples from the patients, and these two genome sequences exactly match with each other. Okay, so going back to the definition by WHO, airborne transmission is defined as the spread of infectious agent caused by the dissemination of droplet nuclei that remain infectious. Yes, we proved that it's viable. And when suspending air over long distance, yes, it's more than 2 meters. We were able to get it even from 4.8 meters away. We didn't get it to do the time study. And the airborne transmission of SARS-CoV-2 can occur during medical procedures that generate aerosols. Well, we proved that even if it's not an aerosol generating procedure, just the patient breathing, he did not talk too much. So even under that condition, we were able to collect the virus and viable virus. I also want to point out the importance of the environmental sampling, as Professor Barberi mentioned earlier. And the reason is conventional phylogenetic network analysis, which is very important in telling us the evolution of this, the virus in different places as a function of time. However, all these samples were from symptomatic patients, very sick, and they go to clinic, so you get these samples. But then you miss the asymptomatic and mildly symptomatic individuals because they don't go to clinic, they don't do the testing, so you won't have their samples. And by environmental air sampling, this is a non-invasive sampling method that will give you, the samples from the entire population, not specific population. So that's what I would like to point to the audience over here. And here, this is our publication. I do like to acknowledge our co-authors. Everyone contributed importantly to this important work. So finally, I'd like to have a summary of my presentation. So infectious SARS-CoV-2 was isolated from air samplers placed 2 to 4.8 meters from COVID-19 patients within their hospital room. And this was detected in the absence of healthcare aerosol generating procedures. And the strain detected in our aerosol sample perfectly match the virus strain isolated from the patient with acute COVID-19. And this system, we really like this system because it very efficiently amplifies the virus containing particles, and that allows effective collection and very importantly, better preservation of the viability for infectivity analysis. I didn't have time to present a different study that we conducted, and over there, the environment sample we collected contained 4 different types of respiratory viruses, and SARS-CoV-2 was a minor one, and therefore it was challenging to really isolate that, although we would detect it, but we were not able to get the viable one over there. And that was in a health clinic. And so for this study that I just presented, we did it in the COVID-19 patient room. And over there, you can say, cleaner environment because only the SARS-CoV-2 virus was there. So we were able to isolate the virus. And then I also want to point out, which echoes others' points, the physical distancing by 6 feet or 2 meters alone would not be helpful in an indoor setting. And this provides a false sense of security and lead to exposure and outbreaks. And I also wanted to emphasize again the genome sequence of air samples, not just the human specimens, allows phylogenetic inference using environmental surveillance instead of a human specimen. And here is the list of additional publications using VIVAS. We have done with the environmental samples, with H1N1 and other viruses. And finally, I acknowledge the financial support from the National Institute of Environmental Health and Sciences and also National Science Foundations, because without the funding, we would not be able to do the sampling and analysis. Thank you. That's all I have for today's presentation. I'll be more than happy to answer any questions. So let me go to the Q&A. How long did you sample with the VIVAS and the BioSpot? Okay, that's a very good question. So in this study, this specific study, we did it for 3 hours, and the samples were good, and the viruses were still live after 3 hours. I would not use other samplers for 3 hours, but for VIVAS, because of the water continue condensation, and therefore you can preserve the viability. And also, but at the same time, the collection medium is also very important. So that's the key to the success. If you use an impactor, if you use a filter, and the desiccation would not allow us to do the viability analysis. Probably RNA is okay, and that has been done by a lot of researchers around the world, but viability is our focus over here. So that's very important to us. Is there any other question?
Thank you very much, Professor Wu, for your presentation and the presentation of your research results with this focus on infectious SARS-CoV-2, which was found in air samples in hospital rooms. So it's a very important information. Thank you so much. I see that there may be some other questions.
I have a question,
yes?
If I can. Direct question. First of all, thank you very much, Dr. Hu, for your fantastic presentation and very nice results. I just would like to know, why did you choose day number 10 to detect the virus?
Not specific 10. We actually had a day 4, day 7, and day 10. Yes, so it's a time series.
Okay, it's a time series. Well, I was
yes.
Figuring out it was. And what about the condition of the cells after 10 days? I mean, they were stable, viable. We are wondering about this topic when working in our small lab, you know, to how to extend the time of infection. So your experience will serve as a guide for us.
Thank you very much for this important question. And I cannot answer the question directly because it's done by my colleagues, John Lennicke's group. And I know that they do it up to 30 days, and after 30 days, they will dispose of the cells. And I think it depends on the concentration of the virus. And if you have a higher concentration, I mean, like within 2 weeks, then all the cells were dead, infected, dead. But if it's a low concentration, like the lowest one we saw on our the slides over here, and then 10 days we start to see the infection, but 2 weeks is not a deadline for a termination of these cells yet. So it depends on the concentration. I think just like what you presented in your presentation earlier, it's a function of exposure time, function of the amount of the virus. But for the details, I have to connect you with my colleague.
Okay, thank you very much. Thank you very much.
And there are 2 questions. Should I answer them now? Okay, so the first one,
Yes.
do you think it's so hard to isolate a viable SARS-CoV-2 in comparison to influenza, for instance? I think it depends on the scale. Again, I'm not the one who did the isolation, and John Lennicky told me that their group does it all the time. It's not an issue to isolate the viable SARS-CoV-2, and there are more studies that we have not published yet, and we have been able to isolate SARS-CoV-2 virus, not just in hospital room, but also in other places. And of course, for the patient human specimens, he has also done that a lot, constantly, daily life. That's his daily job. So if you need more details, then I would connect you to him. Okay, so that's the answer to the first one. And then has this kind of a sampling ever been attempted in crowded areas to see what's the environmental concentration in a non-hospital ward? Okay, so this is again a very good question. I think in order to do this, we wanted to do this, but in order to do this, you need to get the approval from the places. And for example, I tried to get approval from airports, and I always got declined. And actually, one time, one airport said, if you ever get close to our airport, I will ask the police to get you out. So that's the challenge of the getting the sampling in crowded areas. I would wanted to do it in, say, subway stations, but I haven't been able to do that. Now, I can release one piece of information. So we did the sampling in fitness centers, and we did a lot of sampling, and boy, they have done an impressive job. And to maintain it very safe, we were not able to get any SARS-CoV-2 virus aerosol samples in their facility, but they have the ACH of 12, and they also have installed the UV disinfection in the system. It's hard to detect any virus over there. But on the other hand, we have isolated the viable SARS-CoV-2 in isolation room, the self-isolation room in the student housing. That's all I can say right now, but not in crowded area, not easy to get access. That's a limitation. Okay, was the sampling performed in the presence of the patient? Yes, so 2 patients were there. Patient 1 was admitted just 1 day before, and the patient 2 actually got discharged in the afternoon. And this is a very important question because I did say that we were lucky because if the patient had been there for 10 days, I don't think we would be able to get the samples. And the current understanding is that the discharge of the virus aerosol peak at the time of the symptom. And likely after 7 days, you are not able to get any viable SARS-CoV-2 aerosol. You can still detect a lot of these RNAs because these RNA, they are the viruses are produced, but they are also attacked by our antibodies, and they are wrapped by a lot of things, but they are already dead. So you are collecting these dead bodies of viruses, but so you can detect them by RNA, but viable virus, no way after 7 days. at least to our knowledge. Okay. Okay, I think that's all the questions. Oh, did you study the relation with the aerosol size range? That's a good question. And for this study, no, because we did not classify by size. But in other studies, we have. And there is one study that we haven't published yet, and it's everywhere. You have the submicron, and you also have, say, 4 micron particles that have a viable virus. Okay, that's all. All the questions in the Q&A.
Thank you very much, Professor Wu. You answered all the questions, so many questions. Okay, we are on time. I think that maybe Professor Crovella said that he had to go because he has another meeting. But if the other speakers and attendees have still some minutes for the final discussion, if some of the attendee wants to raise his or her hand, we can give you the word so you can speak, so we can have a discussion. Instead of writing, you can speak. So if some of the attendees want to raise the hand, and also maybe there were some questions from Professor Crosta, maybe Francesca and Professor Crovella answered also in the writing form, but if they want to add something, I think that we have some minutes.
I've just go. So I do thank really everybody for the invitation and for sharing the results. A very impressive workshop. Thank you very much. Professor Barbieri is taking the questions for us since we are working together. And I'm sorry, I have to go in my very long day for another meeting. Thank you very much for your invitation and for the collaborative effort. Thanks so much. Bye-bye to everybody.
Thank you. Bye-bye.
Bye.
I would like to ask a question of the panel, in particular the speakers from Italy. What is your national approach now to addressing the airborne transmission? Is this widely accepted, and how can we as scientists, I guess, help inform our elected leaders so that we can have better responses?
May I answer? Try to answer. So we look really for some consensus and occasion to match the different experiences, because there are several different experiences that should be somehow harmonized, and to frankly say consider strong point and weak point. I think this is still missing. A lot of research is done with, say, not within a unified, say, framework. So we are still at this stage. I would really appreciate if we could start some technical norms for harmonization of methods that take into account this, say, the relevant issue that we have faced during this symposium. I mean, there are some researchers say, I haven't found anything in the air, and I have not found anything viable, even less. But there are also evidence that show that in a specific situation we have the problem. So the point of classifying or categorizing, simplifying, structuring somehow the different cases that you can have and to address with proper, say, technological approach, up to date, up to the science state of technology. It is what is needed. I mean, consensus is what on methodology should be addressed, and so I will try to stimulate colleagues both in scientific and technological community for what concerns technical norms, which are the easiest leverage for probably identify some solid or appropriate way of facing the objective. I appreciated a lot Professor Wu, who stated clearly the objective of the study. Very often there is very loose, have a look if there is something in the air. So what? How can you find it? Also, my mother is not able to find SARS-CoV-2 in the air because it is not addressed properly. The question is not addressed properly. So the first thing is scoping. Define the scope of our efforts. And one word was stated in my title. It was interdisciplinary. I'm reading a lot of literature, and I see that top-level scientists are saying it is very highly needed that aerosol scientists enter in the epidemiology circle that define methodologies. So we need to be present and to put our face in front of the other, showing the strong point of our approaches, and then discuss. This is my way. Sorry for not having answered.
Very good. Pat, I see that there is a question for you. If Pat, you want to answer, there is a question by Angela Pizzol about some application of Viva's BioSpot.
So I don't know that I can answer this question specifically because we are the instrument manufacturer and it is our customers who are doing the actual field applications. We have had one customer doing some sampling in an operating room, and in fact, they didn't find anything. But that I think is because of its very clean nature. Isolation rooms, with operating suites are often very highly filtered. And so I think that is a challenge. But as Dr. Wu pointed out, that it's, sometimes you get lucky and sometimes, I think we as a community need to really address this issue of where to sample, when to sample, as well as the how to sample. And I think these are all important considerations. And so I would welcome Dr. Wu's comments because he's done a lot more actual environmental sampling. So.
Oh, thank you. Well, I don't really have any other applications for this device because right now I just focus on the virus aerosol. But in terms of what we can do to better our entire society in defending public health against COVID, I think a very important point aside from technological advances, scientific understanding, or I think very important is the public acceptance of what scientists do. Because very often I have the tools, but I don't have access to do the sampling, where I think the transmission occurs. Because is a hospital the most dangerous spot? I don't think so. I think it's like a bars I think it's the student fraternity party house, but we don't get in, we don't get access. So you can use epidemiological studies to say, oh, it's there, but there is no real evidence that, hey, you have the viable SARS-CoV-2 aerosol over there, because no one is allowed to get in to do sampling. And similarly, in other places, even in public spaces, how can you get access? And this is related to the public perception and public acceptance of what scientists do. And a lot of times, these places, they are very afraid of optics. What if we find a virus over here? Are we going to close it? If I have a virus aerosol detected in restaurant, am I going to close all the restaurants? How about airport? I'm going to close all the airports. And maybe that's the reason they don't want to allow us to do the sampling. But at the same time, without knowing what exactly goes on over there, and maybe it's really safe, like the hospitals. And actually, we have sample in different rooms in hospital, different places in hospital. Most of them we could not detect the virus because they have done a very good job. But if you don't allow people, like scientists like us, to do the sampling to help you identify if it's safe, then how can you really prove that? So that's my point. Thank you.
Okay, thank you. I see that on the chat there is a question for all the speakers about masks. If someone wants to answer about filter material for masks. The question is, what is your opinion about masks? We can see a broad range of different masks, and we know that a lot of them are useful for retaining micro-sized aerosols. Do you think that a better check of filtrating materials for masks should be adopted?
I would say that...
Non scarponi? Ah sì, 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 ce li ho.
With entering, maybe Francesca? So I would say about mask that, of course, if you go for FFP2 or FFP3 or N95, you have some relevant, of course, protection. In some cases, chirurgical masks are not protective for incoming, as it is well known. About the material, there are several studies on this. I don't catch the point. There is a commercial characterization and technological characterization of the mask. We know there's a range of, say, efficiency in blocking different size of aerosol particles and thus viruses. But microbiological, say.
Yeah, hi, this is Francesca, sorry. So it's a great idea to develop new technologies, new fabrics to improve the filtration rate. Definitely, we should also tell people how to use the mask and consider the fact that humidity, when we talk, when we speak, I think many of us have been talking an entire semester with mask on and they get wet. And so then how much do they filter? How many times do I need to check? I need to change them. So giving this information, at least I'm talking I'm referring to the Italian scenario and landscape that I experience, would be really very useful to have a guidance, one voice, and not many drama queens that are just doing their thing. And definitely aerosol scientists can help by estimating the size range of the aerosol, and if there are viruses that of course can infect, but it's overall hard to do it, as we all well know. So yeah, it's up to us to be a good citizen and to be catalyst of the right information, I would say.
Okay. I see there is another question by Francesco Bautiere. I don't know for who, but it's a comment on the possibility of monitoring virus deposited on standard sampling filters recurring to a label. Sorry, the questions disappeared. Recurring to a label-free spectroscopic technique like Raman. But maybe, okay, Pat, did you answer? Okay, you answered this question by the writing form.
Well, I wrote an answer, but it was not specific to sampling on filters. It was specific to sampling with the spot deposits on a solid substrate. And we actually have a couple of different researchers doing that. One that has been published is looking at crystalline silica. And then there is some other research looking at environmental samples and doing Raman analysis. So the concept has been tried. I'm not aware of anybody using it for specifically for biological particle analysis using the spot collection. But I think it's a very interesting concept that should be explored. And in all areas, you know, I think the analysis, different types of analyses can give us different information. And so I think there's not just one way to do the analysis. We need to be exploring which provides the best information for the objectives at hand. So there is an instrument, though, by the way, that it was developed by Battelle that does do collection on a tape. I think it's by Impaction, where they're doing Raman analysis. So, and that was looking for biological particles. I think the challenge with Raman, especially for environmental samples, is really you're looking for the needle in the haystack. So how do you get specificity? How do you separate out the biological particles from all the rest of the particles naturally occurring in the environment? So I think that's the biggest challenge with Raman.
Okay. Did you want to add something, Professor Wu?
Yeah, regarding optical characterization of a virus aerosol, I haven't seen any successful technique. I do agree that this is an important direction to search, but optical responses have been applied for, say, bacteria, fungi, these larger particles with the metabolism mechanisms, but for virus, there has been no success yet.
Okay. So since it's 10 to 6 in Italy, I think that we can close the symposium. So we thank everybody, the speakers, the organizers and attendees for your participation and for your contribution. We hope that you enjoyed this meeting, this event together, and that it was a useful contribution for the community science on bioaerosol research. So, as we say at the beginning of the event, all the symposium was recorded, so the recording will be available in the following days. You will receive an email and you will be able to see the registration, and you will receive an email with the link. So it was a pleasure to share this time with you, and thank again to all of you. And okay, so I can say you goodbye. And I don't know if Pat, you want to add something, and I see that we have also the Professor Stener by University of Trieste, who organized the event with a very good contribution and very precious contributions.
Thank you very much from here, Trieste, and special thanks to these people from US.
Thank you.
Thank you.
Okay.
Have a good day, everybody.
Okay. So good afternoon, good morning in the USA, and good evening in the Qatar. Bye-bye, and we hope we will have other opportunity to meet again together. Okay, we can close the meeting.